Xenopus is a genus of absolutely hideous bloated amphibians from Africa that revolutionized the field of embryology because scientists just couldn't stop messing with these ugly, ugly creatures. Let's start off by learning about their gametes. So the first thing to notice about Xenopus eggs is their pigmentation. The animal pole is darkly pigmented and the vegetal pole lacks pigment. This serves as an important survival mechanism because the eggs look like the ground from the top and the sun from the bottom. There are also two layers of cytoplasm, the cortical and the inner. The sperm can enter any place on the animal hemisphere. The side of sperm entry determines the ventral axis, and the side 180 degrees across will be the dorsal axis. Great to see you contributing to your kid's development, Dad. Once the egg is fertilized, the zygote undergoes cortical rotation, where microtubules in the cytoskeleton turn the cortical cytoplasm 30 degrees with respect to the inner cytoplasm. This exposes a new area in the zygote called the gray crescent, which is super important because it helps set up the conditions for gastrulation. In terms of cleavage, the Xenopus undergoes unequal radioholoblastic cleavage. This means that the entire embryo is dividing, but the yolk cells towards the vegetal pole turn out to be larger and fewer in number than the cells towards the animal pole. Around the 128th cell stage, or the 7th round of divisions, a hollow cavity appears in the blastula called the blastocele, and that's pronounced seal as in the adorable pinniped seal. The blastocele is an important structure because it is a separation between the presumptive ectoderm and endoderm and heralds the mid-blastula transition. Once the MBT occurs, the zygotic genes are activated and the cell divisions slow down. Now we get to the really exciting part, gastrulation. Per convention, we have the ectoderm shown in blue, the mesoderm red, and the endoderm yellow. To start off, some surface cells on the dorsal side called bottle cells begin to move into the embryo. This movement creates the dorsal blastopore lip, and a new cavity called the archenteron is formed, displacing the blastocele. As this happens, the ectoderm moves down and around the embryo in a type of cell movement called epibole. Once epibole has done its job, the blastocele has disappeared and another set of bottle cells migrates into the embryo, and this creates the ventral blastopore lip and lateral blastopore lips on the sides. And the circle of endoderm down there is called the yolk plug. Lastly, the ventral mesoderm becomes mesenchyme, so it can extend and converge with the mesoderm on the ventral end of the embryo. And now look, ta-da, we have 3 tissue layers, each where they need to be on the embryo. Now that was fun, but let's make this hard and go over. Let's start with how the 3 germ layers can be specified with ooplasmic mRNAs. In the egg at the vegetal pole, we have a high amount of VEG-T mRNA, and on the dorsal vegetal pole, we have a ton of VG1 mRNA. Each of these mRNAs is translated into its protein and diffuse in the cell. VEG-T activates the paracontractor nodal, and nodal activates eomes in the presumptive mesoderm cytoplasm, which activates mesodermal VEG-T, starting a positive feedback loop specifying that cytoplasm as mesoderm. On the dorsovegetal side, we have VG1 protein, which inhibits Wnt. Another thing to set up is the beta-catenin gradient. Before fertilization on the vegetal pole, there is a lot of stored disheveled protein. When cortical rotation occurs, this area of protein migrates to the dorsal side. The disheveled is released along with other paracrine factors like Wnt11 and GBP. These factors inhibit GSK3, found throughout the cell, which degrades beta-catenin. The end result of this is more beta-catenin in the dorsal nuclei and less to no beta-catenin in the ventral nuclei. With the VEG-T and beta-catenin gradients set up like this, we can now activate a nodal-related gradient. If an area has high VEG-T VG1 concentration but no beta-catenin, then it will have low nodal-related expression, and will subsequently be specified into ventral and lateral mesoderm. If an area has high VEG-T VG1 concentration in addition to high beta-catenin, then these factors activate Xenopus nodal-related genes, and will subsequently specify into the organizer. Now, the organizer is a super important patch of tissue on the most dorsal, or nucoop side of the embryo. It has 4 big functions. 1. It differentiates itself from the dorsal mesoderm. 2. It dorsalizes the mesoderm around it. 3. It dorsalizes the surrounding ectoderm, inducing neural tube formation. And 4. It triggers the movements of gastrulation. But what do we mean by dorsalize the mesoderm and the ectoderm? Well, the organizer secretes molecules like chordin, noggin, and follistatin. These factors are powerful BMP inhibitors. Along with a bunch of other inhibitory factors for Wnt, this sets up gradients which specify the ectoderm. Where BMP and Wnt are not inhibited, the ectoderm will specify into epidermis. Where there is BMP inhibition but no Wnt inhibition, the ectoderm will specify into trunk and spinal cord nervous tissue. Finally, where both BMP and Wnt are inhibited, this ectoderm will specify into the nervous tissue of the head and brain. And now we've covered the basics of Xenopus fertilization, cleavage, gastrulation, and molecular mechanisms.